Energy system responses. The body contains three energy systems, and an energy system is a system that is made to produce, transform, and supply energy for the body. There are two anaerobic energy systems, the PC store and the anaerobic glycolysis store, and there is one aerobic energy system known as aerobic glycolysis. As you can see in the figure, the PC store is used first in several seconds, then the glycolytic system is used, and once that energy system has been used up, the aerobic system takes over, which is the main energy system, as it produces the most energy. However, the type of system used depends on the sport, exercise, or skill that is being carried out. This section of the assignment will look at this in more detail. The role of ATP in resynthesis. ATP is adenosine triphosphate and fits into nucleic acids. It contains three phosphates, a sugar, which is ribose, and adenine, which is a base. ATP carries energy, and it is required to carry out cellular processes such as active transport or muscle contraction. ATP is gained from food molecules and uses it as fuel for cellular processes. The energy that is used as fuel for the processes is available in the bond between the phosphates, and the energy is released when these bonds are broken. These bonds are broken when a water molecule is added through hydrolysis. When this ATP is converted into ADP, this is due to there only being two phosphates. The sliding filament theory. The sliding filament theory is how muscles contract to be able to produce a force. An example of the sliding filament theory is shown in the diagram. In the sliding filament theory, once depolarization has occurred, causing calcium to be released from the cytoplasmic reticulum, the calcium then binds with troponin, which alters the shape of the troponin, causing the tropomyosin to move off the acting binding sites. The troponin and the tropomyosin blocks the myosin binding site, preventing a muscular contraction from taking place. This means that the myosin heads are able to bind with the actin binding sites to form a cross bridge. This is the important part, when ATP plays a role in muscular contraction. Hydrolysis of ATP occurs at the myosin head before this, so that the myosin can perform a power stroke, whereby the sarcomere appears to shorten as the myosin pulls the actin closer to the center, sliding past each other, known as a muscle contraction. ATP binds to the myosin heads again, causing the cross bridge to be broken, so that when the ATP is hydrolyzed again, the myosin can attach to the actin further down to make the sarcomere shorter. If calcium and ATP is still present, the process will repeat again. Resynthesis of ATP. In order to replenish ATP, there must be energy. Resynthesis of ATP means getting all three phosphates bonded back together. For example, ADP needs another phosphate in order to return to ATP. The reason this is so that energy can be constantly produced in the body. When ADP becomes ATP, this is known as photophosphorylation. If there is no ATP, then the body cannot carry out contractions. This is why the PC store and the anaerobic glycolytic store functions to create energy by splitting a high energy bond, so that this energy can be used to add the P back on to ADP to make ATP. The ATP that is already available in the muscles lasts for around 3 seconds and is used during high-intense anaerobic exercise, such as jumping up for a header in football. A disadvantage of this system is that only 3 seconds worth of energy is produced, which is very little. Therefore, it will not supply much energy during a game sport like football, as it runs out so fast. Although it's advantageous, as it gives energy right away, and it is an immediate energy source. Phosphocreatine store. PC stands for phosphate creatine. The ATP PC store is used during explosive movements, and the rate at which energy can be formed is extremely fast when using this energy store, because it is a very simple process. However, it does not last for a very long time. There is an immediate available store of ATP in the muscles that can be accessed by the muscles straight away when they need it. Once the ATP store of 3 seconds is used in the muscles, there must be a resynthesis of ATP in order to be able to carry on contracting. Phosphocreatine has a high energy bond, and creatine kinase is the enzyme that causes this high energy bond to split. As we know, when phosphate creatine is split by the enzyme, energy is created, and this provided energy goes to the ADP so that the phosphate can be added to make ATP again, with the enzyme of ATPase. The energy from the split of phosphocreatine is needed to make ATP again. The splitting of phosphocreatine is an exothermic reaction, and the endothermic reaction of ATP converting to ATP makes this a coupled reaction. This means that ATP is formed again, and this process creates one ATP, allowing sliding filament theory to take place and a contraction to occur. It is a very fast process because it is so simple and found in the muscles. ATP in PC store creates around 10 seconds of maximal exercise. The available ATP in the muscles lasts for 3 seconds, and the PC store creates 7 seconds worth of exercise. There are certain criteria for when the PC store is used. The intensity of the PC store is very high and occurs when we need a fast release of energy without oxygen. For example, if you were to compete in a sprint of 100 meters, the available ATP in the muscles would be used within 3 seconds, and more energy is needed for the PC store. So the PC store kicks in. The high energy bond is broken very quickly, more ATP is then available through the processes. Therefore, the PC store is highly advantageous for a quick release and need of energy. However, its duration is a disadvantage, as it lasts for a very short period of time, around 7 seconds. This means that it will not be able to supply energy for very long. But another advantage of the PC store is that it has no waste products. But a disadvantage is that it takes 2 to 3 minutes to recover, meaning that you won't be able to use it straight away again. The PC store is used in situations which are at a high intensity and need that short burst of energy really fast. The lactic acid system, or anaerobic glycolysis system, starts with glycogen, which is the stored form of sugars in the muscles, containing 6 carbons, and it is broken down into glucose, and contains 6 carbons from an enzyme called glycogen phosphorylase. This glucose is converted into pyruvic acid with the help of the enzyme phosphofructokinase. At this point, there are 2 times 3 carbons. The process of glucose converting to pyruvic acid creates energy and results in 2 ATP. But at this point, there is no oxygen present still, so lactic acid is created as a byproduct. Leading on, the pyruvic acid breaks down into lactic acid with the enzyme lactate dehydrogenase. Lactic acid inhibits the release of the glycolytic enzymes, which forces the body to stop or slow down, meaning the aerobic system can take over and produce energy in a longer, more complex way. This is because inhibiting the enzyme leads to a high acidity, which the body doesn't like. Homeostasis is what the body wants. The overall total produced is 4 ATP, however only 2 ATP can be used. A characteristic of the lactic acid system is that it's very efficient at producing ATP at a fast rate, and a lot. For every glucose molecule it breaks down, 2 ATP is produced. Being able to quickly produce ATP in a short period of time is very beneficial to athletes, such as in the 400 meters. The intensity of the lactic acid system is high, but not as highly intense as the ATP in muscles or the PC store. The level of intensity decided how long the store will provide energy for. For example, if it was high intensity exercise, the system may only produce ATP that can be used for 1 minute. However, if it was a medium intensity, it may last for 3 minutes. A disadvantage of the anaerobic glycolysis store is that waste products are produced. Lactic acid is produced as a byproduct, which occurs from the buildup of pyruvic acid, as talked about earlier. Lactic acid inhibits the release of glycolytic enzymes, slowing the body down and preventing it from making further ATP. Another disadvantage is that it requires oxygen in order for it to recover, and this process can take up to 1 hour. These are 2 tests showing the changes in blood lactate during a Wingate test and a multi-stage fitness test. As you can see in the diagrams, whilst performing a Wingate test and a multi-stage fitness test, the levels of blood lactate change. In figure 1, at rest, the blood lactate is 1.5 millimoles, however post-test is 7.5 millimoles. This can suggest that whilst the performer is at rest, they are working aerobically and supplying energy to the muscles via the aerobic glycolysis energy system, hence why there is a little amount of blood lactate, 1.5 millimoles. This is because there is almost no intensity at rest and the muscles are not really having to contract very hard. However, once the test begins, the energy system that produces and supplies ATP must change. In the first 3 seconds of the high-intensity Wingate test, the power produced by the muscles will be from the available ATP in the muscles. This is because it is a very high intensity and short duration, and the energy needs to be supplied very quickly. The ATP in the muscles is used very fast, and once again, the energy store changes from ATP to PC store. The PC store is assigned to produce energy for the Wingate test, as the available ATP is used up, and the test is still at a very high intensity, and energy needs to be supplied quickly to the working muscles so that they can carry on working. So the high energy bond phosphocreatine is split by the enzyme creatine kinase, which creates ATP that can be used to join the phosphate back onto the ADP to convert ADP to ATP. Therefore, 7 more seconds of high intensity work is supplied to the muscles. After 10 seconds from starting the test, the energy system must change again, otherwise the muscles will stop contracting. The anaerobic glycolytic system kicks in. The high demands of the test and having to work maximally causes a high intensity. This is why the lactic acid system is recruited. The process starts and glycogen is broken down into glucose, which is broken down into pyruvic acid, which creates more ATP to be used to add the P onto the ADP to make ATP again. However, a byproduct of lactic acid is created. This is why the blood lactate shoots up to 7.5 millimoles in the graph. As the duration of the high intensity work increases, it forces the lactic acid system to work, and eventually there is too much lactic acid in the muscles, and the lactic acid inhibits the release of the glycolytic enzymes, forcing the muscles to slow down in contraction, and therefore less work and intensity on the muscles, to force the aerobic system to work instead. This is why the blood lactate increases in the Wingate test. Eventually, the aerobic system flushes out the lactic acid, and the blood lactate returns to the normal level. This is because the aerobic system is in place and the energy is being supplied by the process of aerobic glycolysis. No lactic acid byproducts are created during this process, so a low level of blood lactate. However, this changes as the test begins, because the performer starts to jog lightly. During the first few levels of the multi-stage fitness test, the energy system used by the performer would be aerobic glycolysis, as it is a very light intensity. As the stages progress, the energy system changes. At level 4, the blood lactate rises to 4.4 millimoles. This is because the intensity has increased, however not enough to switch to the PC store and anaerobic glycolysis. The reason why blood lactate may increase is because there are points when the performer may use the anaerobic glycolytic system, such as if they are a bit late to the bleep, they may sprint just to catch up a bit. This is why there is some blood lactate. As the levels increase, so does the intensity, and there is a point whereby the aerobic system changes, as it cannot keep up with the intensity of the work. In the multi-stage fitness test, the blood lactate level is at rest at 1.5 millimoles. The energy system at the start of the test varies, as all of them are in action. Different energy systems can produce energy faster, or it may take longer. The available ATP in the muscles is a very simple process, and can be used immediately. However, the aerobic system takes a longer period of time to start working, as it is a more complex process. Therefore, at the start of the multi-stage fitness test, the ATP in the PC store is used for the first 10 to 15 seconds. Whilst the PC store is producing ATP, the aerobic glycolysis store is still trying to produce ATP, but the process is taking time. After the PC store is used up for the energy systems, the energy system switches to the lactic acid system. By the process of anaerobic glycolysis, ATP is produced, and only a small level of lactate is produced as a byproduct at this point, because the intensity of the test is very low at the early stage. After around 2 to 3 minutes of the test, and the use of the ATP store and the lactic acid store, the energy system will convert to the aerobic glycolysis, as the complex process of producing ATP by the use of oxygen is completed. In Figure 2, the level 4 of the multi-stage fitness test has a blood lactate of 4.4 millimoles. This is around the time that the energy system switches to the aerobic system. The intensity of the test at this stage is low to medium. As the test progresses, the intensity of the test increases gradually. The aerobic system has to work harder to produce ATP for the muscles to use. Eventually there will be a point in the test whereby the aerobic system will not be able to produce ATP for the high intensity work, so the energy system will convert to the lactic acid system. The lactic acid system will break down the glycogen to glucose and then pyruvic acid with the help of enzymes phosphorylase and phosphocreatine. This will then produce a further 2 usable ATP. However, lactic acid is a waste product of this process.